can actually move on to our biochemistry practice passage. Again, the link is right here, but I'll be posting it along, but I'll also post it again in the Google Doc here if you want to follow along on your computer. I think it's easier a little bit to see. Like I mentioned earlier a little bit, but I want to reiterate again, I'm using this site today. Last time I did it, I'd used Jack Weston. I just want to show you guys all of the free resources that have practice passages that you can utilize when you're studying. There's also a lot of paid ones, but I wanted to showcase some of the free ones that you may easily have access to. So this is one of them. Jack Weston is another. Okay, if you guys didn't know. Okay. Take about, I would say, 5 to 10 minutes. This one's a little bit more complicated and a little more in depth, but still, again, try and keep pace for your timing. About 5 to 7 minutes for this passage. Okay. Okay, let's move on to the first question. Like I said, I feel like you'll be coming back to the passage a lot for this one, and especially to the graphs and just everything the passage, right? So the correct answer is B. Okay, which of the following is the most reasonable conclusion based on figure 3, right? Wild type cells lack adequate levels of ER phospholipids. That's not really supported by anything here. So as you can see, wild type is this dark red. They do have much lower levels of phospholipids than these, but it doesn't mean that they lack adequate levels. In fact, they probably have normal levels. It's just that the deletion mutation actually has high levels rather than adequate levels. So A is not really supported. We don't really have information for that. C, this Sec59-1 acts as a transcription factor to repress lipid expression. That could be one reason, yeah. We can see that when in the wild type, the levels across all of the phospholipids at all time frames are lower than those of the deletion mutation. If you deleted Sec59 and the level of phospholipids went up, one plausible explanation is that it's a transcription factor that represses it. Okay, that makes sense. Sec59 is the primary repressor of phospholipid overproduction. Again, that it represses 59 phospholipid overproduction, which again, because you know that once you get rid of it, you have overproduction. So yeah, that's a plausible explanation, maybe. But for both of these, yeah, it's a plausible explanation, but you don't know that. That's one possible answer, and you have to do way more testing to find out. Again, it's the primary repressor of phospholipid overproduction. Well, how do you know there's not others? How do you know it even is a repressor of phospholipid overproduction? You don't know enough information to say it's D. Same thing for C. Sec59 acts as a transcription factor to repress it. You don't know that it's a transcription factor. You don't know its mechanism of action. You don't know anything about it. You just know that when you get rid of it, you have more. But you don't know how, you don't know why, you don't know anything like that. Just based on this graph. So while C and D are plausible, they're not exactly the right answer. Whereas B, cells lacking Sec59-1 contain higher levels of phospholipids compared to wild type. Very simple, very plain, very easy, just super basic, and that's what you get, right? You don't have to do a lot of thinking. You don't have to make a lot of conclusions. You don't have to make a lot of inferences. It's just based on what you see on the graph, and you just report what you see on the graph, right? What you see on the graph is you have wild type, you have small amounts of phospholipids, you get rid of the Sec59-1, you have a lot of phospholipids, and that goes for all the phospholipids here. Okay, very simple. So you lack 59, you have high levels of phospholipids. Now we'll move on to the next question. Again, this is another graphing question. So the correct answer is C. I think the biggest hurdle with this question is really finding out, there's like so many colors, so many bars, so many everything. What does everything mean? So I think you really have to take a little bit of time to just kind of like find out what everything actually means, right? So this first burgundy, I guess, color is just a wild type. This is just the normal amounts, right? And y-axis again is amount of phospholipid. The x-axis shows which specific type of phospholipid, but really the trends are basically the same throughout for all phospholipids, so the x-axis doesn't matter too much. The y-axis is just the amount, and the colors are really what's important because they're measuring the different variables. This first burgundy color, the leftmost one, it's just the wild type is just what's normally there. Okay. The green is the wild type plus this YEP352 gene, right? And this was what kind of we're looking at, right? And that's kind of like, that's the plasmid, right? So if you have that plasmid, that's what Sec59 overexpression is, right? And I'll go actually back to the passage to make this, to show this even easier. Then overexpression might also have an effect, right? To overexpress Sec59, researchers clone the gene into this vector, right? You guys know what a vector is? It's kind of like a plasmid or something that you, for genetic engineering, you just put it in, and then you into this genome, bacteria, virus, sometimes it's different, whatever. And then you just put it into cell you're looking at, and it'll kind of repopulate, essentially, right? It's a way to just put a gene into something. And the gene that they're putting in, specifically in this case, is the Sec59 gene to overexpress it, okay? So having this wild type plus this YE whatever is just basically means overexpressing Sec59. That's exactly what this green graph means. It's the amount of phospholipids that are present when we overexpress Sec59. Then this next graph, Sec59 with this I delta, right? And you may notice it here. This is the deletion mutation. So this is what happens when you don't have Sec59 at all, okay? And this, in this last yellow graph, is what happens when you don't have Sec59 normally, when you delete it from your normal human person, but, or animal, I guess in this case, but you also put in this vector, which normally would make it overexpress Sec59, but now it just normally expresses Sec59 because now there's only one copy again. It's not technically an overexpression anymore, right? So now that we got that out of the way, now let's look at the levels. You notice how the levels for this graph, and this graph are about the same, with this one being higher. We already knew that and this is the deletion mutation. We know that the deletion mutation compared to the wild type was higher. We knew that from the previous graph we looked at, right? Levels of phospholipids when you delete something is higher, when you delete this mutation specifically, or this gene specifically, it's higher than the wild type. And again, if we look at all the phospholipids, these three graphs are the same, with this one being higher, all the same, this one being higher, again, all the same, this one being higher, so on and so on, right? So now, what is the question actually asking? What effect does overexpression have on phospholipid levels? We know that overexpression is this green graph versus normal levels is this burgundy graph. Just look at it, this burgundy graph, this green graph, they are the same level here, here, and so on. So phospholipid levels remain constant. They only increase in the case of this deletion mutation, which we covered in the last graph as well. They increase in the deletion, but in the overexpression, they actually don't decrease, right? So you'd assume if you delete it, overexpression, if you overexpress it, you would have less levels of phospholipids. That's not the case, as shown by this graph. And D is just like way too much of an assumption. You don't know anything about that, especially since phospholipids gluconeogenesis. You don't really, there's no evidence of that, and it's not shown in the graph. So this one just doesn't make any sense at all. Right? It's really between these three choices, and out of those, C is the correct one. All right. Next question. This one is a little bit more chemistry and physics based. Okay. So, like I said, I don't want to take too much time with this question. It's not really that crucial, but correct answer is C. And the only thing you really had to know was what alpha decay means. Alpha decay is just a helium-4, right? A removal of helium-4. So what that means is you have, I'll use 32p as the starting. Basically means you started with whatever it is molecule started with. In this case, it was 32p. And then it decomposed or decayed into this 4 helium plus something else, right? And first thing you want to look at is the numbers, right? This is the mass, or yeah, the mass. They have to add up, obviously. You can't destroy mass. So 32 goes into 24 plus what? Obviously, then very simple, 28. So you know it has to have a mass of 28. Whatever the product is, it has to have a mass of 28. So this and this are gone. Down between these, now is it phosphorus or is it now aluminum? Well, remember that phosphorus has, I think it's 15, maybe 15 protons, right? It's not written here, but look at it in the periodic table. But really, you don't need to know this question. Just 15 protons. You know, helium has 2 protons, otherwise it wouldn't be helium. The number of protons determines what element it is. So that means 2 protons here. 15 goes into 2 plus how much? Again, you don't lose protons. So it would be 13. And I believe 13 is aluminum, which is C. You didn't have to know. You could have looked in the periodic table. You didn't have to. You just know that it would be another element, right? So the options are phosphorus, which is the same element you started with, or aluminum. Since you know you're losing protons, has to change element to a different element. So you know it's C and it can't be A. If they gave you aluminum or carbon or something like that, then you would have to look in a periodic table which exactly it is to do the math. But in this case, they made it easy for you. All right, so the correct answer is actually A. And a couple things you have to note before we even start the question. Researchers observe an increase in acetyl-CoA in sec deletion cells. Again, what does sec deletion cells mean? Sec deletion means higher levels of phospholipids, right? So why is there more acetyl-CoA why do you increase levels of acetyl-CoA when you increase the amount of phospholipids? Next thing, well, what are phospholipids? As the name implies, a phospholipid is a lipid. It's fat. What does our body do with fats? Well, we went over this a couple weeks ago. Fats get turned into fatty acids, mostly lipids. When they get metabolized, they get turned into fatty acids. Fatty acids then get converted into acetyl-CoA through a process called beta oxidation. And then from the acetyl-CoA, you have a lot of acetyl-CoA per fatty acid, actually, when you do beta oxidation. And then that acetyl-CoA goes into the Krebs cycle, and it just spins and spins and spins, makes a lot of energy, right? That's why fats are really good for giving you a lot of energy, because you can make a lot of acetyl-CoA, and because you have a lot of acetyl-CoA, you can make a lot of cycles of the Krebs cycle, okay? Now, let's move on to the actual question. A, sec deficient cells produce higher levels of phospholipids, which are converted into acetyl-CoA through beta oxidation. That's basically exactly what I just said, so that seems like a good answer. B, sec deficient cells must decrease flux through glycolysis in order to conserve energy. Lipids and glycolysis are not related pretty much at all. A little bit you can have control over it because obviously it happens in your body at the same place. If you're digesting a lot of fats, you don't also want to be metabolizing a lot of glucose at the same time. So if you're digesting one, you don't want to digest the other, or metabolize one, you don't want to metabolize the other, otherwise you make too much energy. But they're really not really connected, and actually having a lot of phospholipids, you would not need to conserve energy. You would actually want to kind of get rid of energy because you have too much of it. So this one doesn't really make sense. acetyl-CoA accumulates as the Krebs cycle stops. Krebs cycle wouldn't really be stopping because you have a lot of energy. If you had energy deficiency, then the Krebs cycle would stop. If you didn't have a lot of lipids, if you didn't have a lot of carbohydrates, then the Krebs cycle would stop because you have nothing to fuel it, to power it. Or if you got rid of one of the intermediates or enzymes in the Krebs cycle, it would also stop. But didn't really mention that you're actually having a lot of fats. Eventually, maybe Krebs cycle will stop because your body will say, I have too much energy, I don't need to make more energy. And it will kind of like put the Krebs cycle on hold. But that doesn't really explain why the amount of acetyl-CoA is increased, right? Because not directly, at least, right? It's acetyl-CoA increases because you have beta oxidation, which then leads to a lot of Krebs cycle, which then leads to negative feedback loop, which leads to less Krebs cycle, right? Which leads to accumulation of acetyl-CoA. But that's like 7 steps away when you have the answer just like 2 steps in front of you. So it wouldn't be C. Hopefully that explanation made sense. And D, cells with Sec59 can only use phospholipids for energy production, causing beta oxidation to occur more rapidly. This one doesn't really make sense at all. Most cells don't only rely on phospholipids for energy production. A lot of them rely on glucose. So that's first thing that's wrong. But just pretend that they were, even then, that wouldn't explain why getting rid of Sec59 and increasing the amount of phospholipids would cause beta oxidation to, like, it doesn't really make sense, right? So it wouldn't really be D. The most, again, sometimes like the simplest answer, the most straightforward answer is the correct one. Like in this case, you just have to look 1 step in front of you. As long as you know phospholipids are lipids, get turned into fatty acids, made into beta oxidation, and you could really get A, right? One of the nice things about the MCAT, it's multiple choice, so you didn't have to even think of this answer if you didn't need to. They give you the 4 answer choices, and you just have to find which answer choice makes most sense, right? So they even, like, as soon as you see A, you might have not thought of it before, but as soon as you see A, you're like, oh, that makes sense. That must be the right answer, okay? And out of these, this is the one that makes the most sense. Here we go. Okay, and so the answer is actually B, right? So yeah, I know Blackstein posted it in the group chat, which is fine. It's not a problem. We're going to go over it right now. Anyway, so YEP plasmid encodes a gene for ampicillin resistance, right? And ampicillin is, you probably should know, is an antibacterial, right? Or yeah, it's just like antibacterial. Researchers treating an YEP transformed bacterial colony with ampicillin should expect what? First thing it should expect, everything. Well, first thing we want to talk about is the YEP we talked about before is linked to the overexpression of, I believe it's, what was it called again? Oh, Sec59, right? So this means overexpression of Sec59, so increased levels of Sec59, right? So what would happen, and it's also resistant to ampicillin. What happens if you had a whole colony that you treated with ampicillin, and this is actually something that's really commonly used, and this is how you get vectors in the first place. You have a whole colony of bacteria. Some of them have the gene that you want, some of them don't. Well, how do you isolate the gene that you do want, which is a Sec59 that's linked, the gene is linked to an ampicillin resistance? Well, you just put ampicillin in. Anything that doesn't have the gene you want, in this case Sec59, will die. Everything that's still alive will have Sec59, pretty much. So what we expect is that the surviving cells will all be having Sec59 or having overexpressing Sec59, whereas all of the ones that are died don't have Sec59, okay? So we're going off that assumption. All of the surviving cells are overexpressing Sec59. Okay, now let's look at the answer choices one by one. Surviving cells have lower levels of PC compared to PC levels in untransformed cells. What did we say about Sec59 overexpression and amount of phospholipids? It doesn't change it, right? We covered that in an earlier question, actually, and that's one of the reasons why I said pay attention to the other question as well. when you're answering a question, think about the questions you already did. So we already know that Sec59 overexpression doesn't lead to decreased or increased levels of phospholipid expression, right? Stays the same as if it was normal, right? So we don't expect the surviving cells to really have any difference in terms of phospholipid amounts compared to normal cells, right? So A would not be right. Surviving cells have similar levels of P and PI compared to P of untransformed cells. Yes, we expect them to have the same levels. Some YP cells to remain sensitive to ampicillin? No, because if they were sensitive to ampicillin, they would have died. So it can't be this one, right? Whenever you treat with an antibacterial, anything that's not resistant to the antibacterial dies. That's just how it is. Acquired resistance to develop rapidly in cells with the YPP plasmid. This is kind of like a weird way to say it. Acquired resistance to develop rapidly. It's not how really it works. Either the genes have the resistance, which in this case they do, and the cells either have the resistance encoded by the gene, or they don't. And the ones that died didn't, and the ones that lived did. So it'd be D. So D wouldn't be D wouldn't be the right answer. And like I mentioned, B is the right answer, right? And we covered most of that earlier already. But with this graph, right? Phospholipid levels remain constant when you overexpress Sec59.